introduce Hans Larsson, our very own Hans Larsson. For those of you who don't know Hans, he holds a Canadian Research Chair in vertebrate paleontology. He joined the Redpath in 2003 as an assistant professor, and he is currently an associate member of the biology department as well as the Department of Earth and Planetary Sciences. He's as of 2011. So Hans's research is diverse and it's very deep, and it asks questions about these macroevolutionary patterns in the expression of particular traits, for example, limbs and so forth. And he thinks of the genetic underpinnings underlying these macroevolutionary changes. So his research is really diverse, and it integrates field work with laboratory work, thinking about what is the mechanistic process under which these evolutionary changes occur. And so without further ado, here's Hans to take it over.
Thanks, John. I'm glad people showed up. I got calls from Ehab, he's at the hospital. I mean, it was a planned thing with his dad, and he's not coming, Virginia's not coming, she's moving house and all these things. Who's going to be here? So before we even get started, I just want to advertise this. It's a new field program that we just got formalized. It took us 2 years, and we're going to run it this year from May through June in Shefferville for the world's first McGill Arctic Field Studies semester. But it's just 3 courses now. Next year, our plan is to hold it on Axel Hyper. So if you have any questions or interested about that, please email me or just email me. So, but for today, it shows up. I'm going to talk about some of the work that we're doing on this topic. So macroevolutionary sort of events, macroevolutionary lives. And these are things that if you think of the origin of insects, or the origin of humans, or the origin of mammals, these are all macroevolutionary events that come with them a whole caveat or whole cadre of novelties. Novelties in the sense of whole gene regulatory networks that are invented, whole anatomical structures invented, and whole behaviors invented. And so, actually, before I even get started there, I just remembered John sent an email a while ago, and while I'm not going to go read the whole email, John in some casual way, I assumed he was on the bicycle at this point. John goes to the gym. I thought he was naturally buff. John watches the History Channel because he saw me on the History Channel while riding a bicycle at the gym. Isn't he more of a Spike TV kind of guy? And I'm super buff and saw your face at the Buffness Center at the gym. That brought me to the threshold to say hi. So, hi, John. Yeah. Now, when you think of buffness, just think John Secant. I don't mean to laugh at myself. So some of the work that we've been doing in the last, I'm just going to say the last 5 years in the lab, it sort of is on this, and I spent some time working on this because this is going to go on our website. And everything in blue is sort of biodiversity, and so that fits nicely into the QCPS funding stream. Thanks, Andy. And where we look at sort of geology and faunal turnovers and climates in the fossil record, and we're describing new plants, new fishes, new marine reptiles, crocodiles, pterosaurs, and dinosaurs, and around the world. The other side, so on the bottom, is more like theory and stuff that's been fun to be involved with an NSF group called Phenoscape. But everything in yellowish greenish is development and evolution, and that's the little pie slice that about half my lab is working in. And in there we work on 2 major transitions. One is the fish to tetrapod transition. The other is the dinosaur to bird transition. And I'm just going to skip along some of the hot topics in both those transitions that we're working on. Some developmental, some anatomical. Just to get into the problems, so this is what the diversity of life looks like. This is the famous Sepkowski curve over the entire Phanerozoic. So it's 600 million years on the x-axis, and diversity in families on the y-axis. Now is on the right-hand side. The issue is that while we can look at ecology and stickleback and guppies and all these things, I'm just sort of rounding off these things that Andrew does. We can look at that to the nth degree, at the genetic level, at the phenotypic level, behavioral level, and all that. The thing is that most evolution is on this macro scale, and macro scale by the order of millions of years. And I once explained to Dan Bolnick that he asked me, so what's macroevolution? We were drunk climbing at Andrew's bouldering gym. And I blurted out to him, well, macroevolution is at scales you just can't imagine. And I have to say, it is at scales you can't imagine. I mean, millions of years. Like, we're not even talking, like the St. Lawrence Seaway wasn't even here or a million years ago. And so we can't even talk about ecology or evolution at the same scale anymore. And so these are the transitions that we work on. So fish-tetrapod, because there's a phenomenal fossil record around it, and there's some really cool ideas in terms of like origin of limbs. And then the dinosaur bird transition, where we have an even better fossil record, and my background actually was all dinosaurs and dinosaur fieldwork. We can think about it, or try to think about it, in terms of adaptive landscapes. And this is something that was really brought into the fold of the neo-Darwinian revolution with George Gaylord Simpson, and has since been sort of been playing around the outside of sort of macroevolutionary studies, some testable, some not. And so what I want to just sort of bring in here is an old image that Simpson brought forth. This is 50, 60 years ago, when he's talking about how to sort of reconcile macroevolutionary changes, in this case, one particular group of mammal-like reptile jumping into another kind of group of mammal-like reptile, and looking like they're going through relative stasis for millions of years. And this stasis with this punctuations in between, we've all heard of called punctuated equilibrium at some point or another, but this is the idea of quantic evolution and a way to sort of skip from different peaks within the adaptive landscape. And so that's why I titled this talk Crossing These Macroevolutionary Canyons, because clearly when you go from one sort of stable state to another stable state in over millions of years of stasis, something dramatic has to happen. And those are the points where I think it's more likely that we're going to come up with some interesting results. Not just looking at sort of incremental fossil records of something that doesn't change, but look for massive scale change. And by this I mean we can look over time on the x-axis here, and if there's some massive scale change in morphology, we can hope to find massive scale change in morphogenesis, and further down gene regulatory networks, further down into codon sequences, and further down into genomic sequences. And once we can actually start to connect the dots between all those different levels in biological, in this artificially, but pragmatically segmented biological research, then I think we can start talking about what really a character is. These things that Dick Lewington was referring to as this quasi-independent unit of evolution, and we all pass around characters as if it's change in our pocket, but I think none of us actually could say what a character truly is, and how to really define one. And so if I were to say what my whole job would be, is to actually find out what a character is. If we talk about the origin of tetrapods, it happened really quickly. So within about 10 million years, we went from fishy-looking things to tetrapody-looking things, completely. And it was a wholesale revolution, going from fins to limbs. That transition came with it, of course, the origin of limbs, so hands and feet with fully formed digits. And there's been a great deal of work, and I'll throw my hat into this a few times, looking at the homology of the fin to limb transition. So how does the forefin relate to the forelimb, and how does a hind fin relate to the hind limb? And so anatomically it's pretty, well, it's still a bit mysterious, but we can sort of look at topologies and say, well, here's the stylopod, here's the zoopod, here's the autopod, and all that. And the problem is that this is where it kind of stops in the fossil record. We can't get any better data than this. We've searched around the world for 100 years, and there are new things coming up, like Tiktaalik, but Tiktaalik looks just like Eosinopteron. And so we're not getting revolutionary new fossils in this picture. One place to look is to take a step back and say, well, here's the fossil record. What about the developmental record? And so on the left is a sort of scala naturae of different fins leading toward terrestrial or tetrapods. And on the right is a sort of a caricature of developmental events that are dependent upon preceding events. And so this, what I was attempting to do here was show that there are sort of developmental things that happen that are dependent on the preceding developmental event. And this becomes very sort of interesting from both a mechanistic point of view, because this is how development works. It doesn't change at all. Everything with limbs develops like this, period. And we can start asking questions, well, how did that sort of mechanism of progression or sort of progress in development, when did that take place? And we can start looking at the anatomy and squinting our eyes and trying drawing lines and come up with these ideas of what I called here a module of developmental evolution. These modes have not taken hold yet in the literature, but they will. But what it is a way to start testing what characters are, or testing what anatomy really is in the context of development and evolution. We can go a little bit deeper than that. So here's the sort of anatomical pattern, and here's the developmental pattern. We can go into some of the mechanistic pattern driving those developmental systems. And what this does here, when we go from, let's say, a coelacanth-looking fin to a lungfish-looking fin, so if I'm right, and Gunther was dragging this too, Roger, if we're right, then we start getting this sort of novel sort of developmental field structuring the Auditorium, or the hands and feet. And if that's going to happen, well, we can go further and say, well, the developmental mechanisms out there predict that Hoxae 11 and A13 have to be involved somehow. And so the reason being that Hoxae 11 and A13 expression is overlapping completely over fin development in all finned fishes, and Hoxae 11 and A13 is completely unoverlapping in all limbed fishes, like you and I. And so probably there's something mechanistic going on here. And if you do complex knockouts for A11, or actually A and D11 and A and D13, you screw up the limb in predictable ways. It gets a little more sexy because Hoxae 11 and A13 are also involved with uteral development. And this was a sort of a collaborative sort of beginning project with Marie Comita at the RCM, where Hoxae 11 and A13 in both uteral development and, it turns out, in the labyrinth formation within the placenta are critical. And so if you tamper with Hoxae 11 or A13 in any placental, you will get completely disrupted pregnancies. So Luke Harrison, who just graduated last year, he's now at McGill Medical School, he left the field. He and I developed this method where we could start looking at DN and DS without having to look at ratios. And this required an amazing amount of work to establish both a sort of a baseline phylogeny based on background evolutionary rates, and then clocking both DN and DS for A11 by itself and A13 by itself. And sure enough, we are coming up with a significant and dramatic evolution of A11 at the fin limb transition, and even more exciting, dramatic evolution at the origin of placentas. So, and not only that, but we've actually localized it to particular base pairs within those genes. Similar things going on in A13, slightly significant at the fin limb transition, and slightly significant at the origin of placentals. But I didn't want to dwell on this, just to show that there are sort of ideas that we're thinking about, and I'd like to talk about all this with anybody later tonight. The research project, though, that I'm going to focus on for the fin limb transition is something that I started a couple of years ago with my postdoc, Anne Stanton. She's now taking a job at Ottawa University. And this was, if we're going to look at this fin limb transition, we can do all we want with development, we can do all we want with genetics. The problem is that it's about 400 million years old, and anything we have alive in terms of the genetic record is going to be at least 400 million years removed from that event. We can think about biomechanics. There's been a couple of really nice papers recently in the last few years in high-profile journals thinking about the biomechanics or the repertoires that some of these fishes or near fishes or fishopods could have gone through. So some of the best models out there, or really the only current models, are from one on the bottom, which is Tiktaalik, and the one on the top, which is Ichthyostega. And both these models suggest that the hind limbs were pretty useless, just sort of very limited in mobility, and that the forelimbs were at best sort of could go through this kind of motion and maybe up and down a little bit. So extremely restricted range of motion, and because of that restricted range of motion, the locomotory gait has been suggested to be something like a seal walking on land. So if you imagine a seal doing like a symmetrical gait, flipping forward with its front fins and sort of dragging its body behind it. So that's the current model for the origin of tetrapod locomotion. If that doesn't ring bells in terms of like that's just absolutely crazy, it should, because seals do it and they're totally derived and really bizarre animals. And why is it that we should get these fishes and salamanders and all these things going through this phase or this transition of locomotion that is just totally unheard of? Let's get this guy here. Well, I'll stay on it. So the origin of tetrapods lies within sarcopterygian fishes. So these are lobe-finned fishes that have these big meaty fins on the front. This is Sauropterus. And some of these fishes got to be massive, so 10 meters or so in length for some of the biggest ones. And so a 10-meter fish crawling on land doesn't seem to really fit. But if we forget about the 10-meter ones and go down back to the smaller ones, there are potentially some similarities in these guys in terms of the lobe finniness and their general shape that make them not too far of a stretch from actually getting onto land. Things like having a ventrolaterally positioned pectoral fin, which is pretty unique for fishes, but these guys have it like right on the ground, for instance. Another issue with the fin-limb transition is that not only do hands and feet come in place, and as things are walking on land, but something really dramatic happens in the shoulder, and this is not really being discussed in the literature. The issue being that basal fishes, or most fishes, sharks, ray fin fishes, all these guys, have a very complex shoulder girdle. So these are all the different fin rays fitting into the glenoid, so the scapulocoracoid. So these are multiple cartilaginous or bony elements that fit into kind of like a peg or dimples on the side of the scapulocoracoid, which really limits the fin range of motion. So if you look at sharks swimming, they don't do much more than this. And you look at most ray fin fishes swimming, they can do mostly this. So they can actually they can do a little bit of abduction, adduction, but there's no fish out there that can actually fully do this, pronate or supinate. That just doesn't happen. When we get to land, the glenoid, or the shoulder joint, becomes very complex. So there's like a saddle-shaped joint where there's a convex and a concave surface for the humerus to slide around and do a lot of complex motion in. If we sort of take a step back and look at fish evolution and think, well, if we have the fossil record and the best we can come up with is a seal kind of locomotion with the fossils, can we look beyond that? And well, of course, lungfish, dipnoans here, are right beside tetrapods. And so it seems like a good place to start. After lungfish, we come to Latimeria, the coelacanth, which lives at 200-meter depths or greater and feeds nose down and has never seen land unless it's brought up in a net or a hook. So Latimeria is just not part of the question, I think, in terms of experimentation. But we can look at dipnoans. And so here's a dipnoan, here's a lungfish. We put it on land and look for what it might do. And it's absolutely pathetic. It's not walking, it's sort of slamming around, and after a little while it gives up. It actually doesn't go forward or backwards, it squirms around and then conks out. It gets lactic. So lungfish, not a good example, or not a good analog for the fin-limb transition. And in comes Polypterus, and so this is where we focused a few years of work on. And Polypteriformes are probably ray fin fishes. Genetic-wise and anatomical-wise, it's actually quite tenuous. And so I think that if someone definitively stands on their soapbox and says that Polypterus is a ray fin fish, I would take that with a big grain of salt. Polypteriformes could be basal sarcops, or they could be basal osteoclenes. They're just so bizarre in terms of their anatomy and so unlike any other fish out there. But what they do have is a lot of plesiomorphic characters, or a lot of plesiomorphic anatomy. And so here's some micro-CT work we've done. Here's the forefin, and you can see right away this forefin doesn't look like a ray fin fish. It doesn't have these elongate lepidotrichia and these tiny little bones and muscles on the inside, but it's got a big meaty pad, very similar to things like coelacanths, very similar to things like Eosinopteron, rhizodontids, Tiktaalik, all these things that have these big meaty fins and little radials sticking out the side. Their head anatomy, I'm not going to talk about today, but what we're discovering is that their heads are even more exciting. They actually have a neck. They can bend their heads up and down, which no fish can do, except for polypterids and lungfish and us, tetrapods. They have lungs, paraventral lungs, so they can breathe fully in the air. They can actually suck air through their spiracles and breathe 80% of their gas exchange is actually through their lungs. They have these ventrolaterally based fins, and the closest are Arcturigians, and it turns out that they can survive on land for some period of time. And I'm going to have a little example here. Right here. So this is a little guy, one of our experimental ones. So this is, so I'm just going to let it go and see how similar he looks to lungfish. And he's never done this before on the stage, so. Do you see his fins? So, but he can live on land for a while. So here's what we're calling gate 2. So if you didn't see on that, on top down, here's what is inside you. And it's actually picking up his entire body off the ground, stilting. I think I can do this again if I just do that. Stilting on each fin and walking forward. And we filmed these guys at 200 frames per second with 2 cameras that are frame locked and then digitized different parts of the body to try and look at where the different fins are going in 3D space and the head's going in 3D space in the body and so forth. There's an even cooler gate, this we're calling gate 3. And if you look carefully, it's actually there. It just flipped on its fin. So it's taking its fin, putting it forward, and as it's coming over it, it's actually turning it over to the other side as it's going. So it's going through a complete pronation and complete supination on its fin. Totally bizarre for a fish. And we weren't expecting this. We were hoping it, but we weren't expecting it. And it turns out if you put them on a very rough substrate, so here at big pebbles or relatively big pebbles, they can actually snake through it. And this is kind of the ground state for fish walking on land, fish moving on land. Trout can do this even. So you take a big fat bodied trout and put it on land in a pegboard, and this was done in the mid-50s, hardly ever sighted. In a pegboard, the fish will actually swim through the pegs. And so the swimming motion on rough surfaces is no problem. But fin-assisted supportive terrestrial locomotion, or what we're calling fast locomotion, is totally unique for polypterus. And what these are, I'm not going to spend too much time trying to explain what a polar plot is, except that each step cycle starts here and goes to here, like for one full step motion, and then back to here. So imagine a line turned into a circle, and then just take lots and lots of steps and put them on those circles. The middle one here shows just a few variables that we were recording, and these variables are things like the start of the fin plant, end of the left fin plant, right fin plant, head, maximum head elevation, minimum head elevation. Gate 2, that one I showed first, has a very sort of tight bars, and what this means is that there's very little variation going on when it's walking. And in fact, the kind of polar distribution that all those different variables have for polypterus walking are nearly identical to those of salamanders walking. So here's a fish that can actually walk like a salamander. This is the gate 1 where they're sliding across the ground, and everything's just random. There's very little variation, or very little. Well, there's just super high variation. So this is kind of expected for a fish flopping on land. This is something that's totally unique for fish, and it's walking like a salamander. The other one is when they're actually flipping their fins over, and it gets complicated. How could they do that? So we got through and just revised the anatomy for these guys. The anatomy, here's the shoulder girdle on left lateral view, so the head is facing to the left. Anything in sort of white and gray and tan is bone, and anything in blue and this yellow here are muscles. And these are muscles spanning the shoulder fin joint. So think of them as shoulder muscles. Here it is in cross section. There's at least 7 different muscles in these fins. If we compare this to things like sharks that have 2 muscles, most bony or most ray fin fishes have 2 or sometimes 3 muscles, and this guy's got 7. Not only does it have an amazing muscle anatomy, but it's also got a very complex glenoid anatomy. So here's if you strip off all those muscles, Here it is in right lateral view. So we're looking at the shoulder this way with the fin coming out the back. The glenoid is actually a ball. So it's not like our glenoids where we have a ball and socket, where the sockets are on our body. These guys have a ball on the body and a socket on the fin. And while that seems bizarre, it turns out that both lungfish and coelacanths have the same arrangement. Only those guys. So here's another piece of evidence suggesting that maybe Polypterus is not a ray fin fish. It's got this reversed glenoid joint. Here it is in cross section. So here's the ball coming off the scapulocoracoid, and here's the fin wrapping around it as a nice cup. If you look on it straight on the back, so now we've taken off the fin, looking at it in posterior view. In blue here is, and done a section through it. In blue is this big basin where a lot of adductor musculature fits into, and this big ligament sort of rides in the back. And so this is a basin behind the shoulder that is probably helping a lot to move the fin around and to anchor it. And Standard will be continuing this now with EMG work in Ottawa. And here's that big sort of convex glenoid in red facing back. So this is what Polypterus looks like. It turns out that we've re-examined coelacanths and lungfish, and they have a remarkably similar anatomy in terms of their skeleton. So here, if we look at the phylogeny, maybe I shouldn't explain so much, but here's Polypterus, here's lungfish, coelacanths, here's lungfish, here's a couple of falciforms, Tiktaalik, Acanthostega, and the living salamander, Necturus. The anatomy for Tiktaalik was introduced as something really bizarre. So here's this normal-looking convex glenoid, which they said was, well, here's the tetrapod glenoid. They said here's this accessory glenoid on the front, and this can only be said for Tiktaalik because it's one of the best things preserved out there. And this accessory glenoid is sitting right on the front, and it's actually convex. And it's sitting right in front of a concave glenoid. And so what the authors for this paper suggested was, well, this accessory glenoid for Tiktaalik is something brand new for Tiktaalik and only Tiktaalik, and it helps it sort of support its fin in some kind of a robust stance phase. What we're suggesting is that this reverse glenoid that the Polypterus has, that coelacanths have, that lungfish have, may actually be just turns into the accessory glenoid. It's sitting in the right place anatomically, and it's also convex, and it's right in the front. And this basin behind, which all the adductor muscles are fitting into, and this strong ligament to hold the fin in place, then maybe just expands, and the humerus slips into it and forms this expanded, sort of complex saddle-shaped glenoid. And if this is the case, then it helps to better explain the sort of radical invention of the tetrapod shoulder as just a sort of incremental evolution of what fish already had, based on this kinematic model. Not only that, but we've looked at the musculature. Okay. We've looked at the musculature in great detail across different fishes. So sharks on the bottom, zebrafish, amia, here's Polypterus, latimeria, or coelacanths, lungfish, and tetrapods. And most of the muscles shown here, all the light gray muscles, are the ones spanning the shoulder joint. And so while we have very simplified muscles in sharks, fairly simplified muscles in most ray fin fishes, with the exception of these erector dorsalis muscles, which actually pull on the leading edge of the fin and let these ray fin fishes do something pretty incredible. Polypterus and up have the unique ability to actually pronate and supinate their fins. So we're calling these rotational fins. And if we were to start sort of drawing sort of major biomechanics models here, I would say that Polypterus allies nicely with Sarcopterygians and having these sort of rotational kind of fins, very different from the ray fin fishes that have erector-driven fins, very different from the very simplified sharks and other chondrichthyans with a sort of up-down fin, sort of like a barn door fin. So we're getting a little bit closer in terms of trying to generate or understand models for this. What this is telling us is that Polypterus and all these other, well, Polypterus sheds light, maybe it's not even Sarcopterygian, but it sheds light on the idea that maybe some Sarcopterygians, before the origins of hands and feet, could actually locomote terrestrially. They could do this fast locomotion. And not only that, the very bizarre tetrapod sort of corkscrew-shaped glenoid may actually be an incremental derivation of what fish already had, this sort of convex and concave glenoid, just sort of evolving into the tetrapod condition. We've done an experiment to try and go a little further on this, to look at developmental plasticity. And this is a paper that we're submitting Monday. All the other stuff is in review. The paper on this paper, we took a group of about 100 Polypterids, these guys here. We got them just when they were babies. Put them right here. We took 100 of them, put them in water. They were the controls. We took another 100, put them on land with little IgA produce misters, keeping their gills wet. And we kept them like that for about a year. And so they grew and lived on land in a very moist condition for an entire, nearly a year. So these fish can live for extensive periods on land as long as they're not too dry. And this is work by Trina Du. And what these are, so after micro-CTing lots and lots and lots of these little guys, both the controls and the experimentals, then this is now laid into sort of standard sort of shape space with size taken out. All the surfaces were modeled with something called spherical harmonics. So rather than putting landmarks on it, Trina just lofted whole manifolds on top of it, and then the manifolds can then be calculated with whatever number of points you want on it. I don't know how many thousand points Trina used for this, more than 100 for sure. And then each of these points can then be looked at across this whole morphospace, and then this is the average map. Anything in color, yellow, is better than 95% significance, and anything in red is better than 1% significant in terms of differences between the controls and the walkers. What this means is that there's a great deal of change, even though it's subtle, and we probably wouldn't have picked it up if we had just done landmark analysis. There's a great deal of change going on in this bone, which is the clithrum, and a great deal of change going on in this bone, the clavicle. This top bone, the supraclithrum, actually in the walkers, or the terrestrialized fishes, retains a very juvenile or very sort of neotenic kind of state, where it's very slender and smooth and doesn't develop all the sort of bells and whistles that normally develop in that bone. This is this joint right here in sort of anterolateral view. This is the control, where the clithrum and clavicle loosely fit on top of each other and they kind of slide around. This is these are the developmentally plastic walkers that now have a tightly fixed clavicles and clythra, and the clavicles even develop sort of novel ridges, get more elongate. This whole shelf on the clythrum gets sort of tucked in. Some really amazing anatomical responses, probably just for living on land. These guys have now also been preserved to later do muscle physiology on them. Is it white muscle or red muscle, so forth. And this figure is still in production. But essentially, if we look at evolution on the left here, so we go from Chirolepis, which you can't see, to Eosinopteron to a tetrapod, this is the shoulder girdle in sort of front or sort of anterodorsal lateral left view. So you're looking down at the shoulder. There's an elongation of this bone, the clavicle. There's a reduction of the top bones in terms of dislocating from the skull. There's also a tucking in of the clythrum away from the whole gill chamber. And if you squint at it, from the controls to the walkers, similar trends in anatomical changes are going on in Polypterus. And so we're not saying that Polypterus is the be-all and end-all, except that its developmental plasticity is showing us the same kinds of trends that are going on this macroevolutionary event. And Pigliucci once said, a few years ago, understanding the full extent to which phenotypic plasticity has influenced macroevolutionary patterns and the origin of phenotypic novelties, like the fin and limb, in particular, may come to represent a major breakthrough in evolutionary biology. And so this, I would say, is the first example so far, that's what first means, of looking at an experimentally derived developmental plasticity that may correlate to a macroevolutionary trend. So this is what Polypterus does, and if it can do it, possibly all these other sarcopter regions can do it when they were being exposed to being terrestrialized, and maybe they responded in the same way. So this is just a developmental plasticity 400 million years later that we're still able to pick up. And we only saw it, and it's relatively cryptic because normally polypterids do not live on land for a year. So we're able to sort of push that plasticity by just forcing them to be on land. In the last few minutes, I'm just going to zip through a bit on our work on the dinosaur-bird transition. Of course, it has a lot to do with flight and the wholesale revolution of birds. If we're going to look at these guys genetically and embryonically, we've only got these guys to work with. So cheetah, turtles, sphenodon, crocodiles, and birds. So, and then many other things, of course, too. But all these guys don't look the same. They're totally different from each other. I mean, the closest living outgroup to birds are crocodiles, and they certainly don't fly. They don't do anything like birds. They do chirp them. And we have this amazing fossil record leading up to it. And the fossil record has also a very nice sort of evolutionary ring to it. I mean, everyone has heard of Archaeopteryx. Everyone has heard that Archaeopteryx came after the origin of species, and Darwin, Huxley and Darwin, all these people were basically picking up Archaeopteryx and say, see, you believe me now? And since that time, we now have, this is the 10th specimen, we now have 11 specimens of Archaeopteryx, and we have a whole slew of feathered dinosaurs in between. Just to impress on the slew of feathered dinosaurs in between, this was state-of-the-art back in 1999, my advisor before Serino. So this is kind of the fossil record as we knew it about 15 years ago, and this was kind of the anatomical record as we knew it 15 years ago. Alex Suchecki and I put together this tree. This is only carnivorous dinosaurs. This is where Archaeopteryx fits. And without reading the names, I think you can see that there's an impressive number of fossils that are filling in these amazing gaps leading up to birds and then post-birds. And so we have an amazing data set to work with. Hundreds of taxa that are beautifully preserved, some with perfect feathers, some with pigmentation on them, everything. The problem is we don't know a lot about timing because we have a fossil record and fossils are everywhere in the fossil record, so we can't get a lot on it. And so Luke Harrison and I took a step back and thought, well, if we're going to be talking about the origin of birds and evolutionary rates and tempos and modes and all that stuff, we need to know what time means. And in the fish world, in the fish-tetrapod transition, it's so old, the fossil record is so poor that we just can't get a good handle on time. Birds we can, because we have 10,000 species either running or flying around us today. And there is actually a 10,000 species tree out there that Yates and it all put together. There are two competing theories, or a few, but at least two major ones. One is that birds originated mostly post the end Cretaceous mass extinction, and one is that most birds diverged pre the end Cretaceous mass extinction. And we need to solve this first. We need to figure out how extant birds have evolved if we're even going to hope to try and bring those evolutionary rates to help clock the fossil record rates. And so what Luke did was went through all the available data out there, assembled more, and came up with greater than 60 kilobases of data for 450 taxa of birds that covered pretty much every major lineage of birds out there. The differences between this and the Yates et al. Is that we're actually running one single analysis with an amazing supercomputing cluster behind it, as opposed to the Yates et al., which sort of concatenated a bunch of individual analyses and stitched them all together and said, voila, here's our tree, which is actually untestable because you can't test the evolutionary rates of any gene on that tree because it's all being concatenated from different apples and oranges. So this is the tree that Luke put together, and this is 40 core years of computing time on both Quebec clusters. And these are 10,000 trees summarized into an MCC tree, just to give you a feeling for the quality of data. And we only picked the best 10,000 out of this, and we picked 10,000 because we wanted to show all of the, or as much of the phylogenetic uncertainty, both topology and branch length, that we were willing to accept. we weren't going to put up one tree and say this is it, because the next tree is going to be different. And so let's just pick 10,000 and show them all and do all calculations on 10,000 trees. And what this shows, though, quite nicely and quite robustly, and we can say this because we've now gone through the most amount of data possible, the most amount of computation possible, and all major bird orders originated in the Cretaceous. We only have a fossil record for one taxon they gave us, which is the duck. But with all the genetic sort of firepower that we could put on this and computational firepower, we're able to say that all orders of birds originated in the Cretaceous with a high degree of confidence, and that the radiation within each order was primarily post-Cretaceous, so post-extinction. So we can now have a sort of a two-phase model of bird evolution. Now that we have this tree based on sort of housekeeping genes, which are expected to be more clock-like, we have a tremendous clock power to start pulling down evolutionary rates into the fossil record. And this is phase 2 ongoing right now. And the full goal of this is to have 250 million years of evolution from the origin of dinosaurs to now, using birds to clock evolutionary rates for everybody in that lineage. So here's our first stab at it. Just using the extant birds, we picked a data matrix. This is Livesey and Zusi, which is almost 3,000 discrete morphological characters by 150 taxa, which is about 500,000 data entries that mostly Brad Livesey did on his own in Carnegie. 500,000 hand-entered data cells for all these things. And this is by far the most complete, the most exhaustive anatomical matrix out there. And we took it, and again, this took several core years to run, but we can now use the genotype, the sort of background housekeeping genes, to clock rates across time. So this is each of these sort of, imagine a vertical bar here that has 10,000 dots on it, and then just a heat map of that going through time. The red is a mass extinction event. The genetic rates are sort of going up and down through time, and now we're able to start clocking on that using genetic rates as our standard, how phenotype is evolving through time. And while it looks a bit messy here, this is because there are actually all branches plotted at the same time. If we do look at isolated branches, we can get some really dramatic flux in evolutionary rates. And so now we're able to really calculate evolutionary rates of phenotype without having to do a sort of a tree based on anatomical data and then back calculate, or in a circular format, evolutionary rates. Now we actually have the tree, which is sort of divorced of all phenotype because we're using housekeeping genes, and then we can throw any phenotype we want onto it. We can also throw any gene we want onto it. So let's say we want to throw a developmental gene, like sonic hedgehog, for instance, to pick something out of the hat. And then we can look at evolutionary rates of particular genes through time based on the background rates of this massive sort of clock that we've developed for birds. For clocks, we sort of entered in this foray. So here's let's look at that. We start looking at anatomical rates of evolution for non-even theropods. So the steeper the axis here, the higher the rate of evolution. So time is on the x, numbers of changes on the y. And so this is the kind of approach that we now want to start layering into our clock models to be able to actually calculate, like, what is the rate of evolution of x or y, or that whole module, let's say the limb. The ecology of birds comes into question because there's a great debate: is it from the ground up or trees down? And for more than 150 years, this debate has raged, and it's still going on now. So one model predicts that birds should start from the trees down, and the other predicts that they may have started running faster and sort of taking to the air by just running fast. Both come with lots of sort of good reasons for that. Many things glide: snakes, frogs, salamanders, mammals, lizards. Many things are gliding out there. So why not birds too? Very few things that we see out there actually run and fly, except for extant birds. So, and it gets a little bit more muddy. Actually, let's go back. The origin for the ecology of birds. Why is it important? Because if we're going to talk about rates and tempos and modes of evolution, we also want to know where they're coming from and the kinds of characters or the anatomy we're dealing with. And so Alex Suchecki and I decided that let's just tackle the ecology of the origin of birds one more time. Maybe we can solve it. Maybe we see something different than other people haven't. And one thing that we saw that other people hadn't was, well, if birds are going to start flight from the trees down, shouldn't they be living in trees already? And anything living in trees today, arboreal mammals, arboreal reptiles, have extremely complicated and advanced adaptations for that. Things like being able to reverse their ankle joint. Some have prehensile tails. They have short, stubby arms and legs generally, so they can stay close to trunks or branches. Very mobile wrists, very mobile hips, and so forth. Even their knees can bend to a greater degree. And so we went through the literature and museums and looking at lots of arboreal mammals, scansorial mammals, things like house cats are scansorial. They can get into a tree, but they can't come out very easily. And terrestrial mammals. And by definition, anything arboreal lives most of its life and feeds in the trees. So that kicks out a lot of things you may think we're actually arboreal. We did the same for birds. And looking at characters like, here's one character, here's the supraacetabular crest, which lies over the femur as it fits into the socket. This is the London specimen of Archaeopteryx. And this bony ridge helps to lock the femur in a fairly vertical position. So these guys couldn't actually do the splits, because then it's bone on bone. And if any of you, especially the older profs here, know what bone on bone feels like, it probably wouldn't do it for climbing trees. And after running all this, what we came up with was that, so here's all terrestrial mammals, all scansorial mammals, arboreal mammals, and a few weird extinct arboreal things. All carnivorous dinosaurs fit in green here, Archaeopteryx in red, and then ground-based birds like ostrich and rhea and emu, and then some of the earliest birds that were actually flying and had a perching foot, fitting in with these other birds here, generally, or generalist birds. If we just stretch these out onto PC1 only, all carnivorous dinosaurs, including Archaeopteryx, fit nicely with things like horses in their anatomy. So there's not a lot of terrestrial or arboreal-looking adaptations in any of these guys. If we compare them to birds, so actually putting out all carnivorous dinosaurs and Archaeopteryx here, and a few of these Cretaceous birds in place, most of these guys, including Archaeopteryx, fit nicely with ostrich and rhea. So high-speed terrestrial runners, and the other Cretaceous birds fit nicely with things like ground-based foragers like crows. And so I think we can say, based on the evidence, based on the anatomical evidence, that the origin of birds has to be from the ground up. There's nothing arboreal about them leading up to and even into Archaeopteryx. We can look at some other things at the origin of birds, and there's like, well, wings. And so wings are supposed to be this sort of dramatic elongation of the forelimb, and all of a sudden, if it gets big enough, then they can fly. And humerus to femur humerus greater than femur is an idiotic character that is still floating in the phylogenetic world on characterizing the evolution of birds. So when the humerus gets longer than the femur, then you're a bird, which is absurd because there's a massive body size changes. And so Alex and I put together this chart. So each of these, so in black is the sort of phylogenetic backbone, and in the boxes are each of the different branches leading off that backbone. So here are tyrannosauroids, here are carcharodontsauroids, here's spinosauroids, whatever. Probably most of your kids know what those mean. And what we're showing is that there's a dramatic increase in body size through evolution, and then a dramatic decrease in body size through evolution. And so if we're trying to measure ratio of bone X to ratio of bone Y, and we have these like sort of tenfold differences in body size going on, there's going to be some signal just coming out of allometry, which is not phylogenetic. And so we very carefully went through trying to come up with phylogenetically corrected allometries or expected allometries for the forelimb and the hind limb for this transition. So what this is in blue is a 95% confidence interval about the expected forelimb length to body size for all non-avian carnivorous dinosaurs. And in black is what the phylogeny actually tracks as it goes through it. When we get to the origin of birds, the forelimb actually escapes. It comes out of the 95 limit and just keeps on going and going and going. So right at the origin of birds, the wing seems to become allometrically decoupled from body size. The hind limb, we did the same thing, and right at the origin of birds, the hind limb becomes allometrically decoupled from body size and actually gets smaller than expected. And if we forget about phylogeny, forget about allometry, and just like line up 3 guys, Microraptor, Archaeopteryx, and Sapiorinus, which are nearly the same size, this is all to scale. One is not a bird, one is right at the origin of birds, and one is into the origin of birds. You can easily see there, even like at this transition, the wings get longer and the legs get shorter. And so there is something really cool going on at the origin of birds, that sort of breaking apart forelimb and hind limb constraint to body size. This has happened a few places, and when it happens, it's pretty exciting. And one group, Young, Halvorsen, and Günther, put together this model where great apes did the same thing. And so their forelimbs and hind limbs became actually allometrically decoupled right when we get to the great apes, and which is separate from all of the primates. And so this allometric decoupling may have some really very interesting developmental mechanisms going on inside. Those developmental mechanisms is part of a bigger project, which on the top left is our logo now. It's called the Chickenosaurus Project, and this is with a group in Montana and myself, and also in Georgia. And we've been going in at some high res into skeletal development and gene expression, finding out what's actually going on in birds and how does it compare to other reptiles and mammals and fish and so forth. I'm not going to talk about that today. There's just not enough time. But this is, if you're interested in this, please call me, or if you have money, please donate. That being said, we can start looking at some very fundamental developmental mechanisms. And so we can look at proliferation, cell death, migration, and morphogenesis, and all these things we're capable of doing, even in a sort of lowly macroevolutionary lab, because the tools are so easy to use, relatively, if you ask Donald. Well, you say something different. We can start applying some numerical techniques now with embryology as well, which is not, or has not, or some reason has escaped the developmental mainstream. And so here we're taking a little embryo, scanning it, and we can measure it, and also do surface, or strip off surfaces and measure surfaces to each other. And we can start by a simple experiment here. I'm just going to throw it out as an idea. We can just measure, here's humerus length to forelimb length to hand length, or this is Joe Halornis, a guy I saw in Beijing, and plot that compared to chick embryology or any other embryology, just to see what happens. Like, how does the forelimb scale through time, and how does the limb grow through time against a phylogeny? So as through evolutionary time, and we can clock it. So black is the humerus, red is the forearm, green is the hand. There is this pattern. I'm not going to say anything about the pattern except that humerus is bigger, and it's almost the same size. Then it gets bigger, and then it just keeps on going. It's advanced statistics. If you look at the embryos, they're doing something surprisingly similar. And I'm not going to say that this is actually sort of von Varian recapitulation. But if we start measuring these embryos, the behavior, like sort of positive, near, and then positive again, that kind of patterns is looking remarkably like ontogenies, recapitulating phylogeny. So we're just getting started on that. Of course, the anatomy is much more complicated than just humerus length or ulnar length. But if you look deeply, there's like here's, if we just look at the first metacarpal of digit 2, I mean, there's all this crazy anatomy going on this one little bone in the finger. And if you look at any single bone, it's going to be as complex. And so we've taken to really trying to visualize embryos to as far down as we can get using optical projection tomography, micro-CT, the old-fashioned serial sectioning with like digital reconstruction and then manual editing, GUI, Tahara. And so, but with this, we've got like almost perfect data to work with. And we can start overlapping things like limbs. And so here's the wing bud at sort of early stage to late stage, and just keep on going. And so all these limb buds we've sort of synchronized in space. And then we can start developing growth models. So we're calling this a constellation method, where it's like geometric morphometrics, but the mechanics are different, where we can now measure the vectors sort of velocity and direction of growth of the whole surface of this one piece of the embryo. We can do it with the whole embryo. We can do it with anything on the inside of the embryo too. And in the end, what we're creating now is actually like a model of development, as opposed to like snapshots of development. We're now creating a dynamic model of development, which we're hoping to be able to use to explain not only extant animal anatomy, but also the evolution of that extant animal anatomy. So this is the same thing, stripped down to just the vectors, or the vector diagram that we can pull out of that. What we're doing here is creating actually a null model, a nice one. In the last minute, I'm just going to throw out a new idea that we've just started on in terms of development and evolution. And that's that can we use embryology as kind of a filter of evolution to come up with some really fundamental developmental mechanisms. And so while I was on the plane flying to LA to give this talk, I was thinking like someone's got to have done this before. And while no one's done it in the last 100 years, Ernst Haeckel did it, I think, but he didn't explain it in this way. But when he constructed his developmental evolution matrix, what he was, I think, essentially doing was taking all the developmental variation, which is sort of on in columns here, and looking at evolutionary variation on each row, and kind of passing it over to come up with a stable state or similarities. And so what he came up with famously was this phylotypic stage. And so that all embryos within the vertebrate world share this phylotypic stage for better or to a greater or lesser degree. But anyways, I think what he was doing was actually using embryonic variation to filter evolutionary variation. And even if he wasn't, we're doing it here. And I just want to bring into this the idea that there's like sort of very complex interactions going on in the head. So that this is work done at UBC, where different parts, so here's an embryo from top down, different parts of the neural crests in the forebrain region, as they migrate down into the head, form everything in pink, and another part in orange forms everything in orange in different parts of the head. So they're very segregated migratory pathways in development. And in fact, even if you do the crude experiment of chopping off the top of the neuroectoderm from a quail, slapping it onto a duck, letting it grow up, then you get a quail beak on a duck head. So there is actually good developmental sort of consistency with that. But it's crude, I should say. But can we do better? Can we actually use the fossil record? Can I justify going into museums and looking at T-Rex and playing with embryos in the lab to actually come up with something greater than just building a better plastic dinosaur or getting another image of a bird embryo? And so this is work with Trina, and this is now being extended into her PhD, where we thought, well, let's just start in 2D for now. We're not going to go crazy and go 3D yet, but can we do it in 2D? So here's our landmark data set for the skull for carnivorous dinosaurs. This one in Alberta. And this is what the landmark data looks like. So this is the procrusting data and all that. This is generally where geometric morphometrics stops. So we get this and we can sort of plot it in the tree, we can draw phylogenetic lines all through and say, wow, goes over here, goes over there. However, this doesn't come with it a null model. Has anyone ever thought of a null model for geometric morphometrics? And so because, okay, we can do a grounding motion model. The problem is though, with enough grounding motion, the front of the skull can actually end up behind the skull. And so we thought long about this and came up with a penalty matrix that were, this is not the right acronym yet, because it doesn't sound right, but biologically informed grounding motion, where we're actually keeping points in a biological rule space, so that the front of the premaxilla does not end up behind the head. And every time it happens, we have an Einstein-Muhlenbeck sort of model where as it gets closer to violating the matrix, it actually gets harder to approach it further and just go back. So what we can do then is plot through evolutionary time. So here's late Triassic, about 200 million years ago, to late Cretaceous, about 66 million years ago. The full range across the phylogeny with both a simulated model, the null model, in gray through that whole time, and the observed data in black. And each of these are representative of actual specimens in a museum drawer. The simulated data actually goes, it's not a complete circle. If we just had complete grounding motion, it would be a nice sort of oval expanding through time, or a cone. But it's not, because we've limited some biological constraints to it. What it's telling us, though, is that there's places where theropods just don't evolve into. So maybe there's something really cool going on here in terms of developmental and or functional constraints. There's one group, oviraptorids, or oviraptorosaurs, which actually evolved out of the expected range. And to give you an idea, for the front end of the skull, it's actually to the left. So they're totally wild skulls for these guys. And so we're now able to start predicting what are the landmarks that are driving this particular complete shift out of unpredicted space. And so with it come also models of developmental and potentially functional constraints that they might be breaking. So we're able to do that because we now have, I think, a better null model. But didn't want to talk much about that, except that, so here's the variation space again. So on DC1, DC2, so you see how the skull is stretching and changing through time. Well, that's cool. We can then take all pre-bird evolution and say, what is the evolutionary variation on the skull? So if you want to come back to this idea that if Haeckel was actually using variation in development and evolution as a filter, can we do the same thing today without drawing cartoons? And so this is a landmark or a heat map of all variation on the skull over 150 million years of evolution. And so this is how it plots out. It comes with a covariance network. I'm not going to talk about that. We can compare that, so now we need a developmental variation network. And so we take a bunch of chicks. So here's many specimens, some landmarks. Some of the landmarks are the same, or homologous, if we want to call the tip of the premaxilla of a chick embryo homologous to the tip of a premaxilla of a dinosaur. And some are not. And the some are not is just a caveat of having an extant taxon. They don't have the same anatomy anymore. But I don't think that matters, because now we can take the chick variation on the top here and actually transform it. So even though the chick head looks different than a theropod, we can take that head and just sort of rescale it to be a theropod. And that's what it looks like. And so this is not trickery. It actually is very sort of philosophically satisfying, because now in reality, what this chick variation, or the chick's morphospace went into, was like a circle, and theropod space went into a circle, where they're now completely comparable, and we just reverse it back into a theropod space. So we're actually comparing apples to apples now, even though the landmarks are different. And I should stress that this is potentially a way to do landmark-free geometric morphometrics across taxa that don't have completely comparable landmarks. So now let's do the filter. So here's evolution in terms of variation. Here's development in terms of variation. We have some kind of union function. Whatever that is entirely up to us. No one else has done it, so we just pick a number. We actually picked 0.8 and came up with this result. So everything in dark is where variation is happening both in evolution and in development. And everything in light is when there's low variation in development and low variation in evolution. So this is a way to kind of use embryology to filter evolutionary variation. Now let's come back to the original question. Can we use that to come up with something fundamental? Well, there are some shocking or some interesting things that come out of this. There are these like big white spaces, and those white spaces kind of, I'm sort of freewheeling here, but they kind of overlap with those developmental modules that have been sort of hacked apart in embryos. And so what we're showing is that this region, which is both developmentally relatively stable and evolutionarily relatively stable, is fitting into a very sort of nice developmental module of the sort of anterior and rectoderm. And the same thing for this other part here. In fact, all the bones here line up perfectly to all the bones over there. So what does this really mean? I mean, could it be that these zones of high variation actually correlate with zones of, the zones of low variation are actually corresponding to these like fundamental developmental modules as they're streaming into the head? Could we be getting a more accurate picture of developmental mechanisms by looking at embryology and evolution, as opposed to hacking off a quail in rectoderm and slapping it onto a duck and doing crude experiments like that? Maybe we can actually get finer resolution information of developmental mechanisms by just looking at the fossil record as well. And so that's what I'm proposing here. Some spin-offs are that if we can now actually really calibrate how low the variation is on certain regions of the skull is, then any character in that space, we can say, well, those characters are going to be evolving fairly slowly. And any character sitting in these sort of high variable places might be evolving faster. And so we're at the beginning of being able to build maximum likelihood models into phenotypic data. That would be very cool. So next time you go to a museum and check out a dinosaur or anything else, I hope that this sort of whirlwind for the last hour has impressed that there's a few more things to look at out there. Building plastic dinosaurs, while it's fun, is not the be-all and end-all of vertebrate paleontology. And I think that there's a lot more to explore in terms of mechanisms of macroevolutionary change. And none of this happens in the vacuum. Thank you.